Hi, my name is Matthias Sury and I'm the Manager Method Development LCMS at our Bioanalytical site in Zurich, Switzerland. In the following 20 minutes, I will present you some practical examples of biomarker assays and their challenges during method development and validation. I will show you where we failed, but also how we could finally solve these issues. During the last couple of years, we developed several biomarker assays at Celerion, and in each project we were facing different challenges. We observed several times that the concentration of the biomarker is quite low, so that the sensitivity of the assay was not sufficient. In addition, especially metabolic biomarkers show circadian changes during the day and are due to their nature highly unstable. Furthermore, it can be challenging to find a suitable surrogate matrix in which standard samples can be prepared or that the molecule selected as a biomarker does not fit for the intended purpose. It can also happen that the concentration levels of a biomarker are unknown at the beginning of method development, or that all of a sudden a commercial kit is not delivered anymore. And finally, it can happen that no issues were observed during validation of a biomarker assay, but due to lot-to-lot variability of key reagents, study sample analysis might be impacted. All these things happened to us, and I will tell you the story of 4 different biomarker assays in which we had to face exactly these challenges. Our first case study is the hunger hormone ghrelin. Ghrelin levels in human plasma show fluctuations during the day as they increase before a meal. Among other functions, ghrelin is triggering hunger feeling in the brain. As nicely shown in this graph, the overall ghrelin concentration is also increased after weight loss, which might explain the famous yo-yo effect after a diet. In addition, ghrelin circulates in the body in an active, acylated form and in its inactive, desacylated form as shown here on the right-hand side. These graphs already demonstrate the importance of a highly standardized sample collection procedure in the clinic, since the time point of sampling is crucial, especially for metabolic biomarkers. In addition, due to the instability of ghrelin in plasma, samples need to be stabilized as soon as possible after collection. For ghrelin, a combination of protease inhibitors and plasma acidification has to be applied. In addition, samples have to be kept on ice during processing. In conclusion, this means even using the best bioanalytical assay in the world will lead to inconclusive results if the sample collection is not highly controlled and samples are not properly stabilized. So this is the story of the ghrelin assay at Celerion. First, a commercial total ghrelin ELISA assay was fully validated in the range of 336 to 1,600 picogram per ml. No issues were observed during validation, and therefore the assay was used for a first study. However, results from this study revealed that 90% of the study samples were below the limit of quantification. Due to this lack in sensitivity, we suggested to develop an LC-MS method, which should also enable us to separate acylated from desacylated ghrelin, aiming for an LLOQ of 50 picogram per ml. So this is in brief the summary of the developed method. In order to reach the anticipated LLOQ for both acyl and desacyl ghrelin, we set up a trap and elute LC system using an additional pre-dilution before separation on the analytical column, as shown here on this scheme on the right-hand side. Precision and accuracy data of 3 independent runs performed during method validation showed that precision for both acyl and desacyl ghrelin was acceptable, but accuracy suffered a bit at higher concentration levels, especially for acyl ghrelin. This was due to the lack of a suitable internal standard, since we were using acyl and desacyl ghrelin derived from rat at this time as internal standards. However, for the intended purpose, the relative accuracy was acceptable. In order to get an idea of acyl and desacyl ghrelin levels in healthy individuals, we did an in-house blood collection. We went to the doctor for blood donation before breakfast. Then we prepared several plasma aliquots and measured these aliquots in 3 independent runs in triplicate. As you can see in these tables, precision of the 3 independent measurements was acceptable. As expected, plasma concentration levels for both acyl and desacyl ghrelin were low, and we saw that the ratio of the acylated and the desacylated form in each subject was almost 1:1. Based on this validated method, we finally measured 516 study samples in which concentrations range from 50 picogram per ml up to 230 picogram per ml. However, still 27% of the study samples were BLQ using the LC-MS-MS method, but at least we could lower the LLOQ in a way that the actual concentration range of Ghrelin in human plasma was better covered than with the commercial ELISA kit. The second case study that I would like to present you is C4, an intermediate metabolite of the classic bile acid synthesis pathway in the liver. C4 concentration levels can be used as a readout for the CYP7A1 enzyme activity. In several gastrointestinal diseases, such as IBS or Crohn's disease, bile acid synthesis is impaired and C4 can therefore be used as a biomarker. In the actual case, we asked the question if C4 can also be used as a biomarker for non-alcoholic fatty liver disease, NAFLD. Based on available literature, we started to develop an LC-MS-MS method in human serum. Since the C4 concentrations in human serum are rather high, we expected that method development would be easy. However, we realized quite early that methods published don't fulfill our needs. First, we could not produce or buy an analyte-free serum pool in which the endogenous C4 concentration was low enough so that it could be used for preparation of standard samples in the range of 0.2 up to 200 nanogram per ml. Therefore, we had to use an alternative surrogate matrix for the standard curve. Due to the chemical properties of the analyte, we observed solubility issues at higher concentrations, and therefore it was not possible to get reproducible results for the standards. Finally, we found a suitable matrix consisting of a high concentration of BSA and a surfactant, showing a nice linearity compared to a standard curve prepared in human serum. Sample extraction procedure consisted of an acid-assisted protein precipitation followed by a solid phase extraction. After setting up this procedure, we qualified the method in order to benchmark the assay. As shown here in these tables, we could show combined freeze-thaw and benchtop stability, and also between-run precision and accuracy data from 2 independent runs were acceptable. In addition, we had the possibility to measure individual human serum samples twice in order to demonstrate high reproducibility of our sample measurement, as shown here in this ISR graph. So finally, we could address the question if C4 is a suitable biomarker for NAFLD, and the answer is probably not. We had the opportunity to analyze a small number of clinical samples from NAFLD patients and from a control group. One diagnostic tool for NAFLD is the FibroScan. CAP is a measurement for the liver fat content, and TE indicates liver stiffness. These are 2 parameters that are elevated in NAFLD patients, and a cutoff value of 307 can be used to define the NAFLD population. As you can see in these graphs, there is no difference between the C4 concentrations of the group with a CAP value below 300 or a TE value below 7, and the group with elevated CAP and TE values. However, this is only a single time point measurement, and it might be that during progression of the disease, C4 levels can change. So what we can conclude from this case study, that it is advisable to measure representative samples as soon as possible using an exploratory assay in order to see if the analytical method is fit for purpose before an assay is fully validated. Case study number 3 is the peptide hormone amylin. Amylin is co-secreted with insulin from pancreatic beta cells and is deficient in diabetic people. Among other functions, amylin inhibits secretion of glucagon. So here comes the story of this assay at Celerion. At the beginning, we fully validated a commercial ELISA assay, but during analysis of the first study samples, non-parallelism and lot-to-lot variability of the ELISA kit was observed. Later on, the ELISA kit failed internal QC checks of the vendor, and therefore the kit could not be delivered anymore. Therefore, we decided to set up an LC-MS-MS assay. An additional benefit of an LC-MS-MS method is that the endogenous amylin can be distinguished from amylin analogs as shown here in this example. In this amylin analog, 2 serine and 1 alanine are replaced by 3 proline, leading to a significant mass shift that can be detected by the mass spectrometer. First, we started method development with a sample extraction procedure using an optimized protein precipitation. Standard samples were prepared in amylin-depleted human plasma, and a stable isotope labeled human amylin was used as an internal standard. With this approach, we could reach an LLOQ of 50 picogram per ml, and the assay was linear up to 5 nanogram per ml. Quite early during method development, we wanted to test in-house collected samples. Therefore, we went to the doctor, collected blood samples after lunch, and prepared plasma aliquots. But unfortunately, all of the individual samples measured were BLQ, as shown here in this representative chromatogram. This was of course disappointing, but we found in older publications some hints which could explain our results. In our LC-MS-MS method, we are using a synthetic amylin peptide with a molecular mass of 3,936 Dalton as a reference item, and the mass transition is set accordingly. However, the amylin exists as a precursor peptide, which is then enzymatically cleaved to its active form. In addition, all the publications describe 2 putative glycosylation sites at the N-terminus of the peptide. This post-translational modification would lead to a mass shift, making the amylin invisible for our mass spectrometric method. In these publications, 2 ELISA assays are described, which use 2 different capturing antibodies. One antibody was raised against the N-terminus, where the glycosylation sites are, and the other one was raised against the C-terminus. Higher amylin concentrations were measured when the antibody against the C-terminus was used, indicating that this would represent total amylin levels. In order to increase the sensitivity of our LC-MS-MS assay, we therefore tested several antibodies raised against the C-terminus for immunoaffinity purification. Although we tested a lot of different protocols, we could not achieve a higher recovery than 5% for the spiked amylin, and this was of course not sufficient to reach an acceptable assay sensitivity. Therefore, we went one step further. We performed the in silico digestion of the amylin and found a suitable tryptic peptide at the C-terminus. After a blast search against the human proteome, we found that this tryptic peptide is unique and can therefore be used as a readout for total amylin. We used amylin-depleted human plasma for preparation of standard and blank samples, and took also our in-house collected individual samples and spiked them with the internal standard. In order to reduce the complexity of the plasma samples, we performed the protein precipitation followed by tryptic digestion. After a sample cleanup step using solid phase extraction, we measured our samples by LC-MS-MS. And finally, these were the results. With this method, we could reach an LLOQ of 100 picogram per ml, and the assay was linear up to 6.4 nanogram per ml. Screening results of individual samples revealed that out of 26 samples, 21 were measured within the analytical range with an overall mean value of approximately 270 picogram per ml. A representative chromatogram of an individual sample is shown above. The fourth and last case that I would like to present you is the story of the human insulin assay. We have chosen a commercial ELISA kit for validation that is able to quantify human insulin in the range of 15 to 480 picomolar. For preparation of standard samples, human insulin affinity depleted serum was used, and QCs were prepared in a human serum pool containing low levels of endogenous insulin. The assay was then validated according to the PK ligand binding assay guidelines. A summary of the most important validation data are shown in this table. Accuracy and precision was within acceptance criteria over the whole analytical range, including DQC. Selectivity and dilution linearity could also be shown. The validated assay was then used for a long-running study. Everything was fine, and the assay had a very high batch success rate. However, after a while, our sponsor approached us because some outliers were observed when some samples were measured later in another run, but not together with the majority of the samples of the whole profile of a subject. Repeating the measurements of all samples of the profile in one run revealed that the previously measured outliers now perfectly matched into the profiles, demonstrating that stability was not an issue of these samples. Based on these results, we evaluated the assay performance over a period of 18 months. In summary, 523 runs were performed, and in this time, 4 different lots of ELISA kits were used. 7 standard sample lots were prepared in 4 different affinity-depleted serum lots, and 9 different lots of QCs were prepared in 4 different serum pools. In this graph, QC concentrations are plotted, and each dot represents one analytical run. The different colors represent the different QC lots that were used. As you can see, the majority of the run QCs are within acceptance criteria. However, fluctuations are observed over time, leading up to 40% difference comparing, for example, the blue and the orange colored runs. Therefore, we investigated the reproducibility of critical reagents and compared different ELISA kit lots and different lots of commercially available affinity-depleted serum. The same lot of standards and QCs were taken and were measured with 3 different kit lots. As shown in the graph on the left, there was no difference between the measurements of the 3 different lots at each QC level. Therefore, the variability between the ELISA kit lots used in the study was negligible. However, a marked lot-to-lot variability was observed comparing different affinity-depleted serum lots from the same and from different suppliers. QCs of the same lot were measured against standard curves prepared in different lots of affinity-depleted sera, and as shown for lot 2 of supplier 1 and the lot from supplier 2, QC concentrations were out of acceptance criteria compared to the initially used affinity-depleted serum lot. So overall, we can conclude, based on our experience, that processes need to be standardized as much as possible in order to get conclusive results. This starts with the sample collection, as discussed for Ghrelin, and ends with minimizing lot-to-lot variabilities during study sample analysis, as shown for the human insulin assay. If available, representative samples should be tested as soon as possible during method development in order to get an idea if the assay is fit for purpose or not. Based on new experimental data, it might be that the context of use of the biomarker is evolving over time, and therefore it should be expected that the biomarker method may also evolve. And finally, communication between sponsor and the CRO is crucial from the beginning of a biomarker assay method development in order to get as much information as possible about the biomarker, because an optimal communication enables us to develop an optimal assay for the intended context of use. Thank you very much for your attention. In case of further questions, please contact me, and we hope to see you at Celerion in Switzerland.